Good morning, everyone, and thank you for joining us this morning. Today, we'd like to share with you how we study cancer and COVID-19 and the connections between these diseases. We represent the Cancer Cell Map Initiative at UCSF and UC San Diego, and also the Quantitative Biosciences Institute and its QBI Coronavirus Research Group. If you have questions during the presentation, please type them into the chat or Q&A boxes and we'll try to answer them as we go or at the end. With no further ado, my colleagues Mehdi Bouhadou and Patty O'Leary will present the first part of our tour. Oh, first we're gonna introduce ourselves, sorry. So I'm Michelle Moritz. I'm a senior scientist at UCSF.
And my name is Mehdi Bouhadou. I'm a postdoctoral fellow at UCSF.
I'm Patty O'Leary. I'm a research specialist at UCSF.
And I'm Robin Cake. I'm an assistant professor at UCSF.
Okay, so let's get started. Thanks for being here, everyone. So today we're going to be talking about viruses and cancer because viruses have been really important to all of us lately. So I'm going to start with something very simple, which is the question, what is cancer? So cancer is essentially a disease that is caused by uncontrolled cell division. So it's when your cells sort of divide out of control. Normally, for healthy cells, cells will divide to some extent. It depends if you're a child or an adult. But then this process is very tightly controlled. So cells that are no longer needed are told to die. And then only a few cells are allowed to replicate. Whereas in cancer cells will acquire mutations in their DNA, which is something we'll talk a bit more about later. And this causes them to divide out of control. So what do viruses and cancer have in common? This is a picture, a cartoon rendition of a virus on the left, and on the right, a rendition of a cancerous tumor growth in a person's body. And one thing that they have in common is that they both need a host to survive. So viruses need to invade cells, and they need those cells in order to reproduce and replicate themselves. And similarly, a cancer also needs a host in order to survive. It needs blood supply, it needs nutrients, it needs a lot of things to keep going and reproduce itself. And as we'll see later, both viruses and cancer cells take over the machinery inside of our cells in order to replicate. So we'll talk a little bit about that later. And now I'm going to pass it off to Patty.
So how do we study cancer and viruses? Because it's hard to work on the human body directly without having kind of understood the disease more. It can be quite dangerous. So we use a process called cell culture or tissue culture. So that's where we're able to grow cells from an animal or plant, human, we're animals, in an artificially controlled environment. So in cancer, what we can do is we can take cells from a cancer patient and we can extract them, and then we mash them up and we can put them into a plate that contains this media that contains all the nutrients we need. And then we're able to grow these cells in a plate. And so we mentioned an artificial controlled environment. So we want to grow these cells in the same conditions as the cells in the human body. So carbon dioxide of 5%, temperature 37 degrees Celsius or 98 Fahrenheit. I think it's 98. I'm from Ireland, so I love Celsius is the way I go. pH of neutral, 7.2 to 7.4. Then also, as Mehdi just mentioned, plenty of nutrients. So in the human body, you have the blood vessels that carry the nutrients in, but we add these nutrients to this media. So this is one way we can study it, is using these plates and these cells growing in plates, but we can also use that to study viruses. That's something we're going to talk about throughout. Next slide, please. So there's multiple different platforms we can do to grow and to study these cells. We can use different kind of sizes of dishes. We have these big dishes that we can grow millions of or billions of cells, which we often need to do for many of the experiments we do. Or we can use these other plates that have hundreds of small little wells, so we can look at a lot of different variations, such as lots of drugs. If you want to progress, bring the bottom in. And so the reason we have all these options is that we have all these different questions that we want to ask when we're studying cancer or when we're studying viruses. For example, one question we can ask is what happens to the cells if we change or mutate one or many of the cell's genes? Or the second question is, what happens to the cells if we treat with drug X, with a drug we're studying? So here's an example of a figure where we can see on the y-axis how many cells are there in this plate, and we can track it over time to see in the presence of drug or a low concentration of drug or no drug at all, a relevant control that you always have to have. You're able to see how quick these cells grow. And you see those pictures, you see in one of them, no drug, so the cells grow really well. And in the other one, the drug is working, there isn't that many cancer cells. So now we're going to go forward and we've prepared a little video, kind of a tour of our tissue culture lab. And it's kind of the platform we use to study cancer cells and this year virus as well. Hello everybody. Welcome to the Helen Diller Family Comprehensive Cancer Centre. My name is Paddy O'Leary. I'm a research specialist here in the Ashworth Lab. So we're a research centre on the Mission Bay campus, right beside the new Chase Centre Warrior Stadium. Here in the Cancer Centre, basic scientists, doctors, computer programmers, we all work together to answer different types of questions on how cancer develops and how we can treat them. Along with many other projects, the Ashworth Lab work as part of the Cancer Cell Mapping Initiative, the CCMI. We're also involved in the Quantitative Biosciences Institute Coronavirus Research Group efforts to help understand the coronavirus. Today we're going to talk about a very important part of our work, tissue culture, or we call it in short TC. So TC is an approach where we grow or culture many different types of cells. We can do it with plant cells or mammalian cells, or in this case human cancer cells, where we can grow them in an artificial setting, in this case on little plastic plates or petri dishes that you might have seen in microlabs. We grow these cells in a liquid or medium that contains all the proteins and nutrients that a cell might need to happily grow and expand. And then we put them in a large incubator that is set at 37 degrees Celsius, or 5% carbon dioxide and relative humidity, so amount of water in the air of about 95%. That's the same physiological conditions as you would see in the human body. So we have to maintain these cells over time so we can use them for all of our different experiments to study them, to see how they grow and how they respond to drugs. So we want to see if the cells are healthy and look as we expected. So we put them onto a microscope. It's called a bright field microscope. And we can see the cells look as they expected. We can see the structure of the cells. But you can see if you look closely here, that some of these cells kind of grow as clumps. You might think that's one cell, or you might think that's 10 or 20. So what we can do is we can genetically engineer the cells so they express this little protein, a red fluorescent protein in the nucleus, so in the center of the cell. And then we can count them individually. So that's kind of a handy way to look at these cells. So now I'm moving over to what we call a tissue culture hood. So this is where we can open the plates and we can ensure that the cells stay sterile. In this TC hood, it's called a laminar flow. So air blows through this hood that prevents things from the outside getting in. So it's a way of keeping it sterile. So the experiment I'm going to do today is what we call splitting the cells. So cells eventually will keep growing and they'll fill out the whole plate, but then the cells won't be too happy because there'll be no nutrients left and no room for all of the plates. It'd be too packed. It's like hopping onto a bus and then it's just packed. You'd feel more comfortable if a few people get off of it. So what we're going to do is going to take the cells, I'm going to lift them off, and I'm going to divide them and only put a small amount of them back in so they continue to grow and expand healthily. Kind of maybe a relevant way of looking at it is we're trying to get the cells to social distance. Pretty apt for these COVID times we find ourselves. So for splitting the cells, first I wash the cells to get all of the old media off and any dead cells that might be there and gunk off the plates. Then I added this liquid called trypsin. So the cells, they stick to the bottom of the plate. They interact, or receptors on the outside, the proteins on the outside of the cell interact with the surface of the plate, so they stay there healthily. I add a thing called trypsin, which kind of cuts those bonds, lets the cells lift up. And then I add media, some more media on top, and I can wash the cells into suspension. And then I put them into a tube and then into this large machine called a centrifuge. So this machine, I'll put in 2 tubes containing cells, and I'll put it spinning, and this spins, God, this one will spin up to 4,000 revolutions per minute, but so the cells don't kind of explode, I will put it at 1200 RPM. So that's kind of the maximum speed that you can spin these cells that is healthy and doesn't make the cells explode, or lyse is another word we use. So now the next thing we're going to want to do is we're going to want to count the cells. So these cells, like each of those plates that you just looked at, that's a 10 centimeter plate, that could have up to 10 million cells in the plate. We're not going to be able to see that by eye. We're not going to be able to count them on two hands. So what we do is we use a machine to count our cells to get an accurate number of cells. So we know how many cells we're going to put into whatever the next stage of our experiment is. So I'm after spinning down the cells, and I see that that little pellet of cells are resuspended in media. So in the red liquid that we add to the cells to keep them growing healthy, and then I put a small amount of it in this machine called a Vicell. So that machine will pull up 0.5 milliliter of cells. I will add this dye called trypan blue to the cells. You can see the dye coming into it now. And then it's going to run through and take 50 pictures of very small amounts of these cells, and it will count in each of those 50 pictures how many cells it will see in that a 10 microliter volume of cells, and that'll give us an average, and we'll calculate how many cells we have in that tube. Then we'll know for the next experiment how many cells we're going to be putting into the next stage. So there's a couple of experiments we could do down from this. Often what we may want to do is to look at how cells respond to a drug. Maybe we want to look at one drug, or maybe we want to look at hundreds of drugs. So there's different types of plates that we use. We call them microplates. So these plates have a different number of wells on them. You can see here in the left hand side I have this little black plate, which is called a 384 well plate. That has 384 wells on the plate. So technically you could do 384 different, you could fill that plate with cells, and you can add 384 different drugs, and we can study it over time and count the cells over time and see how they respond to each drug. Or if you want to do a larger scale experiment, we have this thing called a 6 well plate, which you can use to do 6 different conditions. So today I'm going to seed 500,000 cells into each well of the 6 well plate. So you're going to see me doing that now in a second. So one experiment we use the larger 6 well plate for is the colony formation assay. So in this experiment we seed very small amounts of cells in the plate, we add drug, and we leave the cells for like 2 or 3 weeks. And then at the end we're able to count how many of those individual cells formed a larger colony. So we stain it and we can count those large blue circles. This experiment essentially tests every cell in the population for its ability to undergo unlimited division, which is one of the features of cancer, where the cell can just grow and grow and grow. So the colony formation assay is great if we're only looking at a small amount of drugs or a small amount of conditions. However, if we want to look at hundreds of different drugs or hundreds of different conditions, we use those 384 well plates that I showed you earlier. These little black plates with hundreds of wells on them. We will add the cells, and then on top we would add the drugs. And then the tricky part is, am I going to go through under the microscope and count cells in each one? It would be terrible to do that. You'd be wrecked afterwards. So instead we have a machine that'll do it for us. This is called the IncuCyte Zoom live cell analysis system. So it sits in the incubator. We put the plate in the machine inside the incubator, and then a couple of times a day it'll take photos of each well. And then you're going to be able to see over time how the cells grow in each well under each condition. Also, if we like, this is a kind of a cool video where you can see it growing over time. But also, say if we want to count exactly how many cells there are, we can use those red fluorescent protein positive cells that we showed earlier, and an algorithm will come in that will count each of those cells in the well. So it gives a really accurate representation of how many cells we have in each well. So then over time, you can actually look at how the cells grow. You can see I've got it and point to a graph here that shows the cells growing over time. Every day these cells seem to double. Well, thanks guys. I hope you've enjoyed our little introduction to tissue culture and hope you learned a lot throughout the Bay Area Science Festival.
All right, thanks for that awesome video. So continuing on, and I just want to remind people to go ahead and ask any questions that come up in the question and answer box, and we can get to them at the end of the talk. So as I mentioned before, you know, cancer cells can grow out of control by acquiring different mutations. And when cancer cells grow out of control, you really need a lot of material, right? You know, when a cell divides, it essentially needs to copy all of its components. So the main components that a cell needs to copy for division are its DNA, RNA, and protein. You probably have heard of this, but just to give people a refresher, DNA leads to the production of RNA through a process called transcription. And RNA leads to the production of protein through a process called translation. Now, a lot of you might think of protein as like what's in meat or something, but proteins are actually these small globular machines that are inside of all of our cells. There's tons and tons of different proteins. And they actually do the work of the cell. So they tell the cell to move, to divide, to grow. There's actually another protein which is responsible for making more protein, and that protein is called the ribosome. And so the ribosomes help in this step, in the translation step. So ribosomes will essentially bind to an RNA molecule in your cell and essentially read it like reading a book, and will translate that information into an amino acid strand. And that amino acid strand will essentially create a protein. And interestingly, cancer will hijack these ribosomes to produce more protein. So it'll make these ribosomes work in overdrive. And that's to get more material, right? Because if you're making double of something, you need double of that material to keep going. So this is one way that cancers, and a lot of anti-cancer drugs actually target this translational process. So coronaviruses are similar in this way. So in the top left here, you see a cartoon rendition of a coronavirus, which is mainly composed of RNA, which is this black stringy thing, protein, and lipids. And so coronavirus will essentially bind to a receptor on your cell surface and thereby enter into your cell, and it will excrete its genome into the interior portion of your cell called the cytoplasm. And then what's interesting is that the coronavirus will actually use your ribosomes to translate its genome into proteins that it needs in order to replicate. And so there are many other steps, and this will allow it to continue along its path of replicating itself and then creating new virions that can then escape the cell to infect a neighboring cell. So viruses actually also hijack these ribosomes in order to replicate themselves. So this is one way in which the cellular machinery in our cells is exploited by both cancer and coronaviruses. So the drugs that we actually a lot of our work is actually in repurposing cancer drugs to treat coronavirus infection. We found many cancer drugs are actually effective against coronaviruses if they target the key machinery that both need for their life cycle. So next we're going to talk about proteins and how we study them. And the main way that we study proteins is using a technology called mass spectrometry. And you can think of mass spectrometry as a sequencer for proteins. So a lot of you guys have heard maybe of genome sequencing through 23andMe or something like that. So mass spectrometry is similar, but it deals with proteins. And it lets you understand which proteins are in your sample and how much of each protein is in your sample. And so Robin Cake, an assistant professor in our lab, is going to give you guys a tour of our mass spectrometry facility.
Hello,
My name's Robin, and welcome to the mass spectrometry facility where we analyze proteomic samples. In here, our experiments all start with some kind of protein sample. For instance, if we are interested in looking at what proteins make a cancer cell different from a normal healthy cell, we would take those two different cell types, break them open, so lyse them, harvest their proteins, and then digest those proteins to smaller pieces called peptides that the mass spectrometer can see. So we take our dried peptides and we resuspend them in acid so that they'll carry a positive charge. Then we mix them really well in a vortexer and spin them down in a mini centrifuge. Then we transfer them into special mass spec files so that they don't evaporate before we can analyze them. The next step is to get our samples into the mass spectrometer. Here we use an autosampler that has a robotic arm that can move to the position on the autosampler grid, and basically it pokes a hole in the top of the lid and sucks up the sample, kind of like a straw. Once the sample is picked up, it is passed through a series of tubes where it flows into a sample loop that safely holds the sample as the instruments get ready. And then once everything is ready, the sample flows from the loop out the tubing and into a column. So this is actually the column. This piece here that looks like a wire is actually a hollow tube packed tightly with resin. And so the peptides are attracted to this resin and they want to stay on it. And then to get them off the resin, we use a gradient of increasing organic buffer. And they'll come off the column based on their special properties. And in this way, we can separate different types of peptides so they come off almost in like these packets rather than having them all enter the mass spec at the same time. And so as the peptides come off the column, they come out of this needle. And actually this needle is being electrified, which does two things. It helps ionize and desolvate the peptides. And it's helping to have the peptide spray off of the tip. So we go from liquid all the way through the instrument to gas. So here we can see an actual sample being sprayed into our mass spectrometer. And this little blue light here means that the instrument is running. And so as the peptides go into the instrument, this is basically what the mass spec is seeing. So you can see these kind of dark black peaks coming off, and those are actually the peptide ions. So here we're looking at total ions over time. And here, this is an MS2 scan. So while we can measure intact peptide ions, we can also have the instrument isolate a single peptide ion, transfer it into a chamber, and then hit it with a lot of energy. And this actually breaks the peptide up into smaller fragments that we call sequence ions. We can actually read these ions as you would read letters that make up a word. And in this way, we can tell the different types of proteins that are, say, in a sample that came from a cancer cell versus a sample that came from a healthy cell. Okay, so let's introduce all of our instruments. So this here is the Q-Exactive. It's actually one of our older instruments, and it's kind of our workhorse instrument. This is in fact one of our older instruments, and because of that, it actually analyzes legacy samples. So samples that have been running on long projects that take months to years to collect are all being collected on this instrument. Our new projects are being collected on our newer instruments. So this one is actually the most high in demand, even though it's our older instrument. So this here is our timsTOF Pro, and it's actually different from the rest of our instruments in a couple ways. Unlike the other instruments, which are Orbitrap analyzers, this one is a time of flight. It has this ion mobility chamber that helps further separate peptide ions. And so we can actually separate those packets further using this ion mobility. Imagine jumping out of a plane. So if you have your arms really close to your sides, you're gonna move faster. Whereas if you're wearing a parachute, you're gonna move slower. And peptides are the same. So expanded peptides move slower and contracted peptides move faster. In addition to our instruments, our room is full of different things that we use to help maintain and keep our instruments running. And so this here is a liquid nitrogen tank. And basically we use it when we're taking the instruments apart to clean them. And the instruments, when we put them back together, need to be completely dry before we bake them out. And so we basically take this inert gas, nitrogen, and we flow it over all of the pieces so that when we put the instrument back in, they don't have solvent still attached to them. We actually have also a liquid nitrogen generator, and that's over here, meaning we actually use this to generate nitrogen for the instrument. And you can actually see these copper wires are what transports the nitrogen from this generator to the instruments. And all of the instruments have these copper wires that are taking our highly purified nitrogen gas, as well as helium gas that we have in tanks outside this room, and bringing it to each of the instruments for their varying purposes that they need gas for. Thanks for coming on the tour of the mass spec facility with us.
Okay, so while mass spectrometry identifies what proteins are present, their abundance, and even provides information about how proteins interact, we also want to have three-dimensional models of proteins to help us understand their function and to help us design drugs for the treatment of disease. So how can we see these protein machines from cells? They're incredibly tiny compared to the other things that we normally view in our natural world, as you can see on this scale here. So we have to use electron microscopy to visualize proteins. And in the next series of slides, I'll take you through the process of seeing and building models of proteins using electron microscopy. So the next slide, I'm going to show you a specific example of a structure that we recently solved of a human protein in complex with a protein from SARS-CoV-2, the virus that causes COVID-19. At the top here on the left, you can see a small, a depiction of a small metal grid that we use to apply our sample for putting into the electron microscope. And then there's a blow up view of that. You see it has all these tiny holes. What we do is we apply our protein sample to that grid, and then we very rapidly plunge it into extremely cold liquid ethane that results in a very crystal clear ice in which the proteins are embedded. We then put this grid into the electron microscope, expose it to the electron beam, and we capture these images on a very sensitive camera. So this is an example of an electron microscope image, and all those little dark dots that you see are individual protein molecules. On a computer, we can then pick those particles out of the images as shown by the yellow circles, and in a computer, we can 2D average them to begin to elucidate the structure. We can further reconstruct a 3D image. Oops. It should, oh. So this is a three-dimensional structure of a human protein in complex with a viral protein. And shown in blue here, we can build a model where we can see all the individual components of those proteins. And in aqua is shown the human protein, in orange is the viral protein that sits kind of up in this pocket of the human protein. So the next slide, I'll tell you a little bit more about this. This viral protein is called ORF9b. It's a protein of unknown function that both SARS-CoV-1 and 2 express. And upon infection, ORF9b goes to the mitochondria, which is, you may remember from your high school biology, known as the powerhouse of the cell. It generates the energy that cells need to grow and divide. And on the surface of the outer membrane of this mitochondrion, there's a protein called TOM70 in humans, shown in green here. And if you click that, it will be a little movie, Medi. And this is a sort of a gateway into the mitochondria. So a lot of proteins have to get into the mitochondria, and they normally bind to that pocket that the viral protein is embedded in here. You can see it in orange. So we really don't understand what's going on here. Maybe the viral protein is blocking the normal interactions in the cell with other human proteins, and it's going to require further study to understand that fully. So now in this video, in the next couple of videos, we'll go through exactly how we freeze the grids and then put them into the electron microscope. And this is Fei Li, a postdoctoral fellow, who's going to demonstrate freezing the grids. To take images of proteins in the electron microscope, we first need to freeze our sample on a tiny metal grid in liquid ethane in order to preserve the protein structure. Fei has filled the outer chamber of this insulated container or dewar with liquid nitrogen to make it extremely cold, -320 degrees Fahrenheit or -195 degrees Celsius. She fills the center chamber of the dewar with ethane gas. The liquid nitrogen causes the ethane to condense into a liquid. Liquid ethane is superior to liquid nitrogen for freezing our sample because it has a higher heat capacity, allowing the sample to freeze so quickly that cubic ice does not form, only amorphous or vitreous ice. Vitreous ice is very clear and so won't obscure the images of our proteins in the electron microscope. Fei must be careful not to burn herself with the liquid nitrogen or ethane. The Vitrobot is used to plunge the sample into the ethane and has controls for humidity, blotting time, and pressure. Fei picks up a grid with the tweezers. You can see the grids, which are the small gray discs in the Petri dish. She'll mount the tweezers with the grid on the Vitrobot. She carefully places the dewar onto the Vitrobot and then fastens the tweezers in place. The Vitrobot will draw the tweezers and grid up into the humidified, temperature-controlled chamber, and Fei will apply her protein sample to the grid through the port on the side of the chamber. The Vitrobot moves the dewar into position for the grid and the sample to be plunged into the ethane. Now Fei draws up a bit of her protein sample and pipettes it onto the grid through the side port. It's just a tiny drop of liquid that she applies to the grid. The excess liquid will be blotted away, driving the protein into tiny holes in the grid where they will be frozen in a single layer in the ice. The sample then plunges into the liquid ethane. The Vitrobot lowers the dewar. Here you can see that the grid is submerged in the liquid ethane in the center chamber. A small round grid box is just visible in front of the ethane chamber and the surrounding liquid nitrogen. Fei will quickly transfer the grid into the box for storage until it can be loaded into the electron microscope. So now we'll see David Bulkley, the director of the EM facility at UCSF, explain one of the state-of-the-art electron microscopes.
The idea with these instruments is we're trying to image things that are super incredibly tiny. So like literally invisible to the naked eye. And the reason for that is the light that we use with our eyes to see everything in the world is, I guess you'll learn more about this in physics, but basically it has a certain wavelength. And that means anything smaller than the wavelength of light that we use is invisible. And so the biological molecules we want to look at, so all the proteins, RNA, DNA in our body, are are so incredibly tiny that they're way smaller than the wavelength of visible light, so you can't see it with your eye, no matter how closely you look. So in order to get at those super teeny tiny little things, what we need to do is we need to use something with a shorter wavelength. In this case, we're using electrons. So we have electron microscopes that have a really short wavelength, and we're able to look at these incredibly tiny molecules. And then at the top, we accelerate the electron. So they're coming down from a source up here, and they're coming down in a really tiny coherent beam. And the beam's going to come down through here, and what these are different lenses and apertures that we use to focus that beam and make it as good as we possibly can to get an image on a tiny portion of our specimen. And our specimen is going to be like right here. After the beam goes through, it passes through the sample, and then there's a lens that reforms it into an image, just like your eye reforms what you see into an image, right? Mm-hmm. And then takes it down to a camera. But this blue thing here is part of our camera that we use. As the beam hits the sample, it's actually going to cause a lot of disturbance to it. So it's going to cause it to move around and actually move up and down in the XY plane. And that's because these electrons that we're using are very high energy. And that causes the image to blur. So what we're seeing down on the camera is not going to be super sharp. It's going to be a blurred out version of what we were looking at. But these cameras are able to acquire images in extremely fast frame rate. So they operate at 1600 frames per second. And that means that as the sample's moving, we can actually capture a movie of it. And if you have that movie, then you can unblur what happened. And this turns out to be super duper critical for getting really high resolution information in these samples. So if we're able to unblur it, we can get all the way down to the atomic details of the protein. So we can see even like individual atoms in a molecule. But then these are just tanks of liquid nitrogen that we have. That's what we use to keep it cold. So this is the camera computer and the microscope computer. And we're able to send commands through the software to the microscope. And these are old samples that we've used. And it's this tiny disc. It's this copper mesh with a layer of carbon suspended on it. This here is like that. So, and then what we do is we go in and we look. So this part is made of copper, and then this is a little bit of carbon. These are little bits of carbon on here. So then we go in at a higher magnification, and we zoom in on like one of these little squares. If we zoom in on one of these squares, it looks like that. And so each of these holes is about 1 micron, so that's a millionth of a meter, so super tiny. And what we do is we set down targets on each of these. So each of these is a position that we're telling it to acquire a target at. So this one, for instance, is empty, and this one has a nice bit of ice in it. And these are large chunks of contaminating ice that we don't want. And then we'll take an image of each of those. And so each of these images in turn is going to look like this. So this image is actually I think it's like 1100 by 800 pixels or something like that. I forget the exact numbers, but it's a very large image, and we're just looking at it kind of zoomed out. So you can go in and take a look at each one of these individual protein molecules, and you can average them together. So the signal from any one of these is almost nothing. So it just looks like a blur, and you can see the image itself looks like a TV static. But once you average it together, then you can get a really clear picture, and you build this three-dimensional map of what the protein looks like.
So we hope you've enjoyed our videos. Basically, our goal is to understand biological pathways in cells, and protein structure and function allows the development of drugs to combat diseases from cancer to COVID-19. And some of you may have received a little freebie that we had associated with our presentation, which are these build-at-home paper microscopes called a Foldscope. I hope they went very quickly. So even if you didn't get one, I encourage you to go to this website and learn how you can make your own paper microscope and explore the world around you using that. So next slide. We have plenty of time for some live Q&A now. And so if we haven't answered your questions, please feel free to ask them. and we'll discuss and elaborate a little bit further on the questions that we've already gotten. Thanks a lot.
So we do have a few questions that are coming in. Steve Soth asks, how long does it take a sample to be run through the processes depicted here? So maybe we can start with cell culture and then work our way through mass spec and then cryo-EM.
Mm-hmm. One interesting thing about cell culture, one fascinating thing, some of these cell lines that we work with have been growing. They were taken out of a patient maybe 30, 40, 50 years ago. But then we put them in the freezer and we can revive them at a later stage. And they're being used all over the world and shared all over the world. But the experiments itself kind of often it depends on the questions we're asking. A lot of our experiments, say for example, we're testing a drug in those plastic dishes, we'll be just putting them on the cells for 3 or 4 days. But then we've some longer ones that are more longer term assays, like I described that colony formation assay. But yeah, experiments can vary from 2 or 3 days through to, say, if it's an experiment with an animal model, can be up to a year. So some of our questions are quick answers. Some are take a long time to get your answer.
I would say the same is true for mass spec samples as well. It really depends on the type of experiment that you're doing and the type of samples that you want to generate. So some samples are pretty straightforward and we're just looking for what proteins are in a sample, what proteins are in a protein. Some of them we want to actually purify the proteins out of a cell and we just want to look at a small subset of proteins, and so they take a little bit longer to generate. If we have to do any type of biochemistry to get more pure samples. And then if you have a lot of samples, it takes a long time to run in the mass spec. And if you only have a small subset of samples, then it's a little bit faster. Yeah, so some of our projects we can get through the entire process in a couple weeks, and some of our projects take a few years to generate all the samples and analyze all the data.
And David Bulkley is here too. David, would you want to describe how long it might take for a cryo-EM process?
Sure, yeah, the sample preparation part's really quick in terms of making those thin little layers of protein on the grids like Michelle and Faye were showing you. So that part just takes a few minutes. But the imaging stage maybe takes a couple days. It depends on what your sample looks like, but the analysis can be quite tricky. So for instance, I collected data on a sample in 2016 actually, and I wasn't able to really get a good answer out of all of my data until around 2018. So it took me a long time of sort of banging my head against the wall and trying to figure out different ways to look at it to be able to analyze and interpret it correctly. And the reason for that is the data that we get is extremely noisy. So you saw the images that we had of those we call them micrographs. So it's like those gray fuzzy pictures with little proteins on them. And there's a huge amount of noise in there. So it's really tricky to be able to generate signal to see to tell what you're really seeing from those noisy images. And that process can take a little while. But I will say that it's improved and sped up a lot recently. So on average for your standard protein, you can know within a few hours to a few days roughly what your sample looks like. And then you have some more time to sort of polish and refine your result. But it can take a very long time. And there's still a lot of things that we're working on now where we really want to know the answer and we still don't. So we're trying as hard as we can to figure it out. But so very variable, as the other folks have said.
Okay, great. We have another question from Tracy who asks, where do you obtain the samples of SARS-CoV-2 for research? I'll take a brief stab at answering this. So some people do clinical research with clinical samples, and they obtain samples through like hospital settings, through participants that have given consent to provide samples. We actually just work with the virus in cells in a culture dish. So the virus has been given to us from a reputable source, and we essentially culture the virus in the lab. And so we keep it growing in the lab and just use it whenever we need to do various experiments. Does anybody have anything they'd like to add to that?
I would just say that the lab where the live virus is used to infect cells is a very, it's not every lab that can do that. There are very stringent controls for safety, and the people who actually do that work are very highly trained in how to deal with the virus. And then I thought I would also add that in terms of getting structural information, like the structure I showed with the human protein in complex with a viral protein, there we are expressing the viral protein through recombinant DNA work in a different cell type. So it's only the individual proteins that are being expressed. And so then it's perfectly safe because we're not dealing with the virus itself at all.
I think one thing to kind of add to Michelle's comment about the kind of the really high protective lab, that lab was only really, as far as I remember, was just set up this year in response to the COVID pandemic. So that's one thing. There was a question earlier on about kind of the international collaboration from UCSF, where groups all across the world. So impressed to see this year how labs all across the world just developed to answer this one question, which has been really impressive, and how much we've learned about this disease. And the QCRG, so the research group, it's interestingly, the research group at UCSF that we're all part of sent reagents to 42 different countries to help hundreds of labs progress their understanding of this disease. So it's kind of just open borders, free give our reagents to everyone, because together we'll be able to understand something better. It was very impressive.
All right, we have another question. What are the connections between multiple sclerosis and coronavirus? And I actually don't know the answer to that question. I mean, I'm sure there are some molecular mechanisms that overlap. As some people know, multiple sclerosis is a disease which degrades a certain white matter in the brain. I don't know if people want to comment on that. I don't really have much insight into that.
Yeah, I'd say multiple sclerosis is unfortunately one of the few diseases that the Krogan Lab doesn't study. But yeah, we're always interested in finding connections between different disease pathways. So who knows, maybe we'll look into that.
Yeah, I think it's a great question, and something that is definitely very interesting for future research to look into. The connections between all kinds of diseases is super interesting.
Maddie, we also had a couple of questions in the chat. So one question was on, there's actually a few questions about the drugs that we're testing. So maybe I'll just summarize. So one of the questions was on if the drugs we're looking at are used to treat COVID rather than prevent, and the answer is yes. So, so far no one is testing or generating drugs as prophylactics, so something you would take before you get the virus. Other than there are people working on vaccines, that just doesn't happen to be what we do. So all of the drugs we're testing are for once you have gotten the virus, it would be to treat COVID. And then there's a similar question about whether the drugs we're testing are currently undergoing additional licensing, are they new, or are they currently available on market? Mehdi, do you want to answer that one since you have the most involvement with the drugs?
Yeah, sure. So the drugs that we're testing range all of these. Some of them are already FDA approved, and so in a lot of our studies, we try to prioritize these drugs because these have the quickest route to being approved for use, since they've been shown to be safe in humans for other diseases. But we also test drugs that are still in preclinical phases or in clinical trials as well.
There was another question in the chat, just so that we don't lose track of all the questions, asking why you can get COVID from breathing in virus, but you can't get cancer that way. And maybe we all went away. So cancer happens when the cells inside of you mutate. And so it's actually a disease that is created from inside of your body. Whereas COVID is caused by a virus, so a foreign body that can be passed from person to person because it can survive outside of the body for a short amount of time. And then once it gets inside another host, it can start replicating again. Whereas cancer is pretty much contained within your own personal body. But Mehdi, maybe you want to talk about.
I think that's exactly what I would have said as well. I think that covers it.
Although I would add that there are some viruses that can cause cancer, like hepatitis C, that was the subject of the Nobel Prize winners this year, who it's very dangerous in terms of causing liver cancer.
There's also, along Michelle's lines, cancers like that are caused by something like HPV, which has oncogenes inside of it that will transform your cells into cancerous cells. So yeah, the viruses and cancer is pretty intricately linked.
I will also add that, so far we don't have any evidence suggesting that coronavirus causes cancer. We don't want to scare people. We don't think it does. We have no reason to think it does, so don't be worried. But there are a lot of similar machinery is exploited by both, which is very interesting to think about.
Ren had a question saying the things demonstrated today appear sophisticated as diverse, and do we need to be jacks of all trades as scientists, being able to do everything? But that's actually the true beauty of collaboration, that we have people who are skilled in all different sets, and we understand one set of experiments to a very intricate depth. And then we work with each other and we hand off projects and let someone who's a true expertise, or an expert in mass spec, in tissue culture, in proteomics, to do the work for us. So that's kind of the beauty of collaboration.
So someone had also asked what other viruses that we work on. Collaboratively, we probably cover a wide range of viruses. At least in the Kruggen lab, we study HIV, we study Zika, Ebola, flu. So we study a bunch of different types of flu strains. We've studied hepatitis virus and HPV virus, so they're human papillomavirus, coronavirus. We study a number of bacteria as well, so chlamydia and tuberculosis. I'm probably missing a couple. I'm trying to think off the top of my head, but we study quite a large number of viruses and bugs, so bacteria as well, and yeah.
Katrin asked, how long has the type of sophisticated equipment we use been available? Some of the machines, I'm sure the mass spec machines might be only developed in the last couple of years, the more modern machines, but then some of the cell lines we use have been used in experiments since the 40s, 50s, and 60s. It's we have kind of a whole range of, like, some of our gold standard experiments to do have been used for decades. But then there's
so actually mass specs have been around for a long time too. Yeah, so mass specs have been around for a long time. They haven't always been used to study proteins the way that we study them. So a lot of times they were used to measure small molecules. Kind of in the mass spec world, similar to computers, you get like a new generation of a mass spectrometer every couple of years. And so like, yes, the newest kind of fanciest mass specs are recent, and the older mass specs have been around for a while. But yeah, they're kind of like computers, and you get a new model every couple of years.
And they get smaller and smaller and smaller.
Yeah, and they get smaller, and new bells and whistles.
Yeah, and exactly the same for electron microscopes. Like, they've been around since I think the 1930s or maybe even earlier. But every couple of years we get sort of like a newer development. And so the change from one to the next is very small and incremental. But then if you look at it over like a 30-year period, you're like, wow, these things have really changed a huge amount. But you might not notice it from one year to the next. But yeah, they're always evolving and developing.
Robin, someone is asking, how is a general mass spec cleaned between samples? It seems with all the tubes and needles, it's impossible to clean it fully.
Yeah, I was just about to answer that in the chat. It's Cleaned very carefully. So we actually don't do a full cleaning very frequently. We usually just clean the front part, which is what I was kind of indicating in the video. And basically we have to shut the whole instrument down. We vent it. So mass specs are under high pressure, so they have to be vented before we can take them apart. And then we have a bunch of little tools that we can take all the pieces apart, and we have really fine sandpaper actually that we use to clean off some of the harder to clean junk. And then they're cleaned in pretty powerful solvents that we use, and something called a sonicator, which applies kind of this really high frequency sound that is trying to clean all of the metal pieces of it. So yeah, everything is cleaned and then dried with inert gas, and then we carefully put everything back together and bake down and pump up our instruments so that they're back under high pressure.
John has a question. We only have 2 minutes left. John has a question. Does SARS-CoV-2 multiply faster than cancer? If so, why? My answer would be that, it depends on the time scale of infection. I think that SARS can take a while to take hold, but then once it has, it can really replicate very quickly. So I would say it definitely has the potential to multiply a lot faster than cancer. Cancer cells also vary a lot in how fast cells divide. So some cancer cells will divide in 24 hours, others will divide in a couple days. Some are slow growing, some are fast growing. So I think it really depends on the cancer type. But viruses can definitely replicate very quickly once there's enough of them around.
Yeah. Viruses are really, really little, and they have a minimal component. And so one cell can actually produce hundreds, thousands of baby viruses. So they have exponential growth. That's why viruses can usually multiply faster than like one cell trying to divide needs all of its materials to be replicated, whereas a little virus just needs a minimal amount of materials to replicate. I also see that a question was asked about what's the difference between COVID and flu, and could this COVID be just a bad flu? So coronavirus is actually quite different than flu. Even though they're both RNA viruses, their genomes are very different. So flu has a segmented genome, meaning all of the pieces of its genetic material are separated. So it actually has each piece, each protein is a separate gene. And then in COVID, it has a continuous genome. So one long piece of genomic material. And it's actually much bigger. So it has a lot of genes, whereas flu just has like usually 8. And they're very distinct from each other actually, in what they look like and what they can do inside of cells. And even for flu, so flu has a lot of differences depending on the type of strain. So yeah, there's quite a lot of differences between the viruses on a molecular level as well. When we study them, we can see how they change cells differently from each other. But we are very interested in seeing what the similarities are to see if we could find some drug or some factor that we could target that could potentially target both viruses, but they are very different.
There's a question from Mark Chang. Is there a hope to eliminate the COVID-19 virus on this earth? I would say yes, but maybe someone else wants to elaborate, or?
I would say yes too, for sure. I mean, I think that we could at least eradicate it in humans. It might persist in other organisms and animals. I mean, it seems to have hopped from another organism to humans, so it definitely will persist, likely persist in those organisms. But I think definitely if we have a good vaccine and if we have treatments and testing and contact tracing, we can certainly, it's certainly possible to eradicate the virus.
Okay, I think we're at our end of our time. Yeah. Close us out.
Okay, well, thank you guys for joining us today. I've really enjoyed it. I think we've all really enjoyed talking to you all. Thank you for being curious about science, and hope to see you next year, hopefully in person.
Thanks, everyone.
Thanks.
Guys.